Greeting and Assalamualaikum warahmatullahi wabarakatuh. My name is Nabill Afiff Abdul, together with my team, Atira Ahmad Fauzi, Sarah Shafiqah Juli, as well as our supervisor, Dr. Liana Ahmad Zawawi, will be presenting the finding on banana peel extract for silver nanoparticles, biosynthesis, and its related chemical compound. So as an introduction, biotechnology involves the use of nano-sized objects range from 1 to 100 nanometer. So silver nanoparticle is one of the most vital and interesting nanomaterial among some of the metallic compounds that are elaborate in many biomedical applications. It has good physiochemical properties of stability, thermal and electrical conductivity, and robust optical feature. So the significance of silver nanoparticles are broadly discussed on their multifunction application, such as anti-inflammatory, bacterial, viral, antifungal, and anti-cancer. It also has been incorporated into consumer products from various sectors such as electronic, healthcare, textile, food and beverages, and water treatment. So the preparations of silver nanoparticle mainly consist of two approaches by physical and chemical method. So physical approach method essentially work with the solid in its bulk form. For example, thermal decomposition, laser ablation, electrical and gamma irradiation. So chemical approach on the other hand contain an identical system which involves catalyst, reducing agent and enzyme as an example that manufacture nanoparticles organized by catalyst asset, reaction media, and condition such as solvent, stabilizer, and temperature. Example is borohydrate reduction, Tollern reaction, polyoprocess, and toluprocess. So, however, both approaches have limitation, which are highly energy consumption, low yield, and produce toxic byproduct. So another method is known as biological procedure, where microbe and plant are used as reducing agent to be in the nanoparticle synthesis. The employment of biological components of silver nanoparticle preparation is known as green synthesis. The main aim for this green chemistry is to protect environment from pollutant. So some of the advantages of the green synthesis include high efficiency, economical, less waste, safer product, compatible for pharmaceutical and biomedical application. So extract from this bioorganism might perform both as a reducing and capping agent in the silver nanoparticle preparation. So this drive by the interactions of metal salt with the functional group found in these agents, which eventually reduce phenolic form of silver ion into quinone form by reducing two electron. So the
Problem statements of this research is
fruit waste are generally utilized and discarded as a solid waste at large expense. So it's significantly essential to find application for this peel as they can contribute to environmental issue. So banana peel, for instance, are rich in compound with great benefits on medical properties, ethanol fermentation, substrate for generation fungal biomass, bioabsorption for heavy metal, and productions of lactase. So lactase is an enzyme for pulp and research paper. So because of its great benefits, we are interesting to prepare silver nanoparticle in greener method using banana peel extract from its biological application, such as antibacterial activity, bioremediation, including algae flocculation efficiency. So the present study aimed to synthesize silver nanoparticle by a green biological route using an extract derived from banana peel waste, while the second objective of this study will be include the identification and characterizations of the silver nanoparticle synthesized from the banana peel extract, or BPE AGNP, right? There are four stages during the research. The first stage is extraction, where solid liquid extraction was used to extract banana peel and pineapple peel. So for banana peel, the peel was dried and grind into fine powder before it was boiled and filtered using filter paper to get the extract. In the synthesis step, BPE or banana peel extract are mixed with a metal salt, which is silver nitrate, and the color of the reaction mixture started changing to yellowish brown within 10 minutes. and to reddish brown after 1 hour, indicating the generations of silver nanoparticle due to the reductions of silver metal ion into silver nanoparticle via the active molecule present in the BPA. The third stage is characterizations of peel extract and biosynthesis silver nanoparticle using instrumental analysis. So silver nanoparticle was isolated from the solution using centrifugation and it was oven dry. So several instrumental analysis was done to verify the successful of BPA in the synthesizing of silver nanoparticle. So for the silver nanoparticle from banana peel extract, UVVs, FDAR, EDX, and FISM are used to characterize, while total phenolic compound or TPC used to characterize the BPA. So to characterize successful formations of silver nanoparticle from banana peel extracts or BPE, AG, and PA, the color changes form after reactions of BPE and silver nitrate was monitored visually and characterized by UV-Vis. So as you can see in the set image in the figure, the color reaction changed from yellowish brown to reddish brown color after 72 hours compared to the silver nitrate alone on the left side. So for the FISM analysis is done to observe the morphology of the silver nanoparticle. BPE AGNP shows spherical shape which agreed with the result from the visible spectra and observed formations of single SPR peak. So for the size of BPE AGNP has size of 29.09 nanometer plus minus 6.5. For the EDX analysis is done to determine the element present in the silver nanoparticle. It can be seen that silver nanoparticle is the main element as high as 88.9%. So there are also presence of other elements in the BPE silver nanoparticle, such as carbon, oxygen, and chloride. So additional element might derive from chemical compound from the extract, right? So the presence of carbon and oxygen elements supported by the FTIR finding. So the FTIR was done to determine the functional group involved in the biosynthesis of silver nanoparticle. Based on the FTIR spectra, we can see that the BPE AGNP has relatively same peak as the BPE. So the significant band appeared at 3371, 1779, 1635, and 1180 that assigned to OH, C double bond O,
CN and CO group.
This shows that the biomolecule in BPE is involved as a reductant and a stabilizer in silver nanoparticle synthesis. So the functional group that is involved during the silver nanoparticle synthesis are hydroxyl, carbonyl, amide, and amine. So the functional group may come from the phenolic compound or other molecule present in the BPA. So the functional group appeared in the FTIR spectra may correspond to the several main components of the banana peel, such as carbohydrate polymer and proteinaceous compound. The example of carbohydrate polymer include cellulose, hemicellulose, lignin, and pectin. So the presence of the single CN or amine spectra in the FTIR can explain the protein compound of the essential amino acid present in the banana peel. All right, so banana peel is known to be rich in dietary fiber, protein, essential amino acids, polyunsaturated fatty acid, which possess various beneficial effects to human health. Other than carbohydrate, polymer, and protein aqueous compound, polyphenol that plays important role for the antioxidant activities may be one of the components to interact with the metal salts and mediate their reduction to nanosilver particle. So this study, we further investigate the total phenolic compound analysis to measure the phenolic content of our BPA. Based on TPC, it was done to quantify phenolic compound in the banana peel extract. So phenolic compound is one of the abundant molecule in the plant which has ability to act as a reductant. So the phenolic content in the BPE is 88.65 GA milligram per gram. So based on this study that has been done, example of phenolic compound in the banana peel extract is palmitate, capritates, xanthophyll, and lurate, which might involve in the formation of silver nanoparticle. In conclusion, the following work has present a quick synthesis of silver nanoparticles from banana peel extract. So the biosynthesis of silver nanoparticle using BPE were characterized as spherical, uniform with average size of 29 nanometer. Several chemical compounds such as pectin, cellulose, hemicellulose, protein, and phenolic group are possible components that involve in the reduction process in the synthesis of silver nanoparticle from banana peel extracts. Lastly, I would like to acknowledge Ministry of Higher Education for the financial aid to support this supporting research. Thank you.